Hematology, red blood cells. The erythrocyte. The red blood cell membrane separates intracellular fluids from extracellular fluids. It selectively allows nutrients and ions to pass in and out of cell. It allows the cell to deform when necessary to squeeze through microcirculation and return to its original shape. Hemoglobin structure and function must be adequate. The membrane is composed of lipids and proteins. The phospholipid in the membrane have a polar end, or hydrophilic, water-loving, and a nonpolar end, hydrophobic, does not attach to water. The lipids, cholesterol, and phospholipids are in the red blood cell membrane and helps regulate the membrane fluidity and permeability to electrolytes and other substances. The amount of lipids in the membrane is affected by the lipid concentration in the plasma. Integral proteins. They start from the cytoskeleton and expand through the membrane to penetrate the outer edge of the red blood cell surface. They are the backbone for active transport. Water, chlorine, and bicarbonate diffuse freely. Sodium, potassium, and calcium are more highly regulated. The sodium-potassium pump. Sodium ions are pumped out of the cell, and potassium ions are pumped into the cell. The energy to drive the pump is released by hydrolysis of ATP. Membrane permeability. Spherocyte membranes are more permeable to sodium. Red blood cells are more water permeable during lysis, and both have a reduced lifespan. The cytoskeleton. Membrane proteins are spectrin and chirin, actin, and protein. These help provide the biconcave shape. Lack of these results in abnormal red blood cell shape and decreased red blood cell survival, or hemolysis. Deformability and elasticity are important properties of the cytoskeleton. Spectrin and ankyrin are responsible for the deformability. If the plasma lipids get out of balance, the red blood cell membrane can become deformed. Abnormal red blood cell morphology. Anisocytosis is variation in size. Macrocytes are over 9 micrometers, and microcytes are under 6 micrometers. Poikilocytosis is a variation in shape. Indices. Mean corpuscular volume, or MCV, is 80 to 100 FL. Mean corpuscular hemoglobin, or MCH. Mean corpuscular hemoglobin content, or MCHC, is 32 to 36%. Variations in these values correlate with differences in color and size. Variation in color. Polychromasia, more blue. Hypochromasia, less color or more pale. In polychromasia, reticulocytes are gray-blue and larger than normal. They show anemic stress, and there is excessive production of red blood cell precursors in response to this anemia. The bone marrow releases reticulocytes and sometimes orthochromic normoblasts, or nucleated red blood cells, prematurely. Hypochromia. Larger than normal central pallor. MCHC is less than 32%. Usually seen when hemoglobin synthesis is impaired. Usually a gradual process. All microcytic cells are hypochromic. Variation in shape. Spherocyte. Dense, dark, and small. Near normal MCV. Elevated MCHC of over 36%. Mechanisms of spherocytosis. Hereditary spherocytosis is inherited spectrin abnormalities. They start normal, and as they negotiate the spleen, it senses the membrane imperfections and shears the exterior membrane. Aging RBCs lose pieces of the membrane as part of natural cell death. Spleen removes the imperfections until death. Antibody coating. Autoimmune or immune response. Antibodies are removed from the membrane by the spleen. Sickle cell, or drepanocyte. Crescent shape and pointed projections at one of the terminal ends. Mechanisms of sickle cells. Hemoglobin S, hemoglobin SS, or homozygous S, has a dramatically reduced lifespan. Hypoxic stress is induced. Sickle cell anemia. Sickle trait, hemoglobin SC disease, and hemoglobin S thalassemia. Stomatocyte. Elongated areas of central pallor. Mechanisms of stomatocytes. Hereditary stomatocytosis, or Rh null phenotype. Chemical agents, alcoholic conditions, poor slide technique, or liver disease. Ovalocyte. Egg-shaped, capable of many variations of hemoglobin distribution and size. Mechanisms of ovalocytes. Macroovalocytes are caused by a megaloblastic process. Normochromic ovocytes are caused by thalassemic syndromes. Elliptocytes are cigar-shaped. Mechanisms of elliptocytes are abnormal spectrin and protein 1.4 component, hereditary elliptocytosis, iron-deficient individuals, and idiopathic myelofibrosis. Target cell or codocyte, bullseye-shaped, bell-shaped, or cup-shaped. The mechanism of target cells could be an artifact, decreased volume secondary to loss of hemoglobin, increased red blood cell surface membrane, increased plasma cholesterol causes surface to expand, hemoglobinopathies, thalassemia, iron deficiency anemia, septicemia, obstructive liver disease. Acanthocyte or spur cell, irregularly spaced projections that vary in width, length, and number. Mechanisms of acanthocytes. Excess cholesterol, increased surface area, and decreased lecithin. Liver disease, autoimmune hemolytic anemia, McLeod syndrome, or lack of KEL antigens, lecithin cholesterol acyltransferase, or LCAT deficiency, congenital abetalipoproteinemia, splenectomy, malabsorption, hypothyroidism, and vitamin E deficiency. Burr cell, or crenated echinocyte. Short, evenly spaced projections. Teardrop, or dacrycyte. Teardrop or pearl-shaped. Mechanisms of teardrops. Myelofibrosis with myeloid metaplasia, thalassemias, myeloproliferative syndromes, severe anemias, myelophthisic anemias, extramedullary hematopoiesis. Fragments or schistocytes. Fragments of red blood cells, many sizes and shapes, often display pointed extremities. Mechanisms of fragments. Microangiopathic hemolytic anemia, hemolytic uremic syndrome, thrombotic thrombocytopenic purpura, disseminated intravascular coagulation, severe burns, or renal graft rejection. Red blood cell inclusions. Howell jelly bodies, siderotic granules or Pappenheimer bodies, basophilic stippling, Heinz bodies, reticulocytes, and Cabot rings. Howell jelly bodies are composed of DNA. They stain with Wright's stain and new methylene blue. They're megaloblastic anemia, hemolytic anemia, splenectomy, and hyposplenism. Siderotic granules, or Pappenheimer bodies, are composed of iron. The inclusions are termed siderotic granules in Prussian blue stain and Pappenheimer bodies in Wright's stain. Small, light purple, beaded inclusions located along the periphery of the red blood cells. Hemolytic, sideroblastic, megaloblastic anemias. Hemoglobinopathies. Splenectomy. Basophilic stippling, composed of RNA, stains with Wright's and new methylene blue. Fine or coarse dark blue to purple granules. Lead poisoning, thalassemia, and abnormal heme synthesis. Heinz bodies, composed of unstable precipitated hemoglobin. Stains with new methylene blue. G6PD enzyme deficiency. Some hemoglobinopathies. Unstable hemoglobin. Kappa rings. Loop, ring, or figure 8. Composed of remnants of mitotic spindle. Stains with Wright's and new methylene blue. Myelodysplastic syndrome, or megaloblastic anemia. Hemoglobin C crystal. Hexagonal shape, composed of hemoglobin C. Homozygous hemoglobin C disease. Hemoglobin SC crystal. 1 to 2 finger-like projections, may look like a mitten. Hemoglobin SC and hemoglobin SC disease. Hemoglobin function and principles of hemolysis. Chapter 4. Hemoglobin and the red blood cell. The function of the red blood cell is to produce, package, protect, and transport hemoglobin among various tissues. Hemoglobin is responsible for carrying oxygen, or oxyhemoglobin, pulls CO2 away from the tissues, or carboaminohemoglobin. It keeps balanced pH between the blood. The structure of hemoglobin. The heme portion is 4 iron atoms in the ferrous state, surrounded by protoporphyrin 9, or a porphyrin ring. The globin portion is 4 polypeptide chains, 2 alpha and 2 beta chains in most adult hemoglobin. Heme molecules in globin pocket. 4 heme molecules with iron at the center and 2 pairs of globin chains. The heme structure sits in the pocket of each globin chain. There are 3 types of hemoglobin in red blood cell production. Embryonic hemoglobin, which includes hemoglobin Gower 1, which has 2 zeta chains and 2 epsilon chains, Gower 2, which has 2 alpha chains and 2 epsilon chains, and Portland, which has 2 zeta chains and 2 gamma chains. Fetal hemoglobin, which begins 3 months after fetal development starts. It includes hemoglobin F, which is 2 alpha and 2 gamma chains. Adult hemoglobin, which begins 3 to 6 months after delivery, includes hemoglobin A, which has 2 alpha and 2 beta chains, and is 95 to 98% in the blood. Hemoglobin A2, which has 2 alpha and 2 delta chains, and is 3 to 5% in the blood. And hemoglobin F, which is 2 alpha and 2 gamma chains, and is less than 2% in the blood. Oxygen dissociation curve. A right shift means that hemoglobin has less attraction to oxygen and is more willing to release it to the tissues. A left shift means that hemoglobin has more attraction to oxygen and is less willing to release it to the tissues. Oxygen dissociation physiologic conditions. A right shift would be caused by anemia, decreased pH or acidosis, increased 2,3-DPG, or elevated body temperature. A left shift could be caused by decreased 2,3-DPG, a reduced body temperature, presence of abnormal hemoglobin or high oxygen affinity hemoglobin, or increased pH or alkalosis. Abnormal hemoglobin is physiologically abnormal. It can produce conditions that are toxic to the human body, and something other than oxygen or carbon dioxide is bound to the iron molecule in the heme structure. Some examples are methemoglobin, carboxyhemoglobin, and sulfhemoglobin. In methemoglobin, the iron in the heme molecule is oxidized to the ferric state, which makes the heme unable to bind oxygen. So it goes from Fe2+ to Fe3+. The oxygen cannot be transported in these red blood cells, and over 10% of individuals will become cyanotic, or blue in color. Carboxyhemoglobin. The heme molecule binds carbon monoxide. It has over 200 times more affinity to carbon monoxide than it does to oxygen. This can cause carbon monoxide poisoning. Sulfhemoglobin is a sulfur atom incorporated into the heme molecule. It is a permanent change and is clinically detectable at 5 grams per liter. It causes organ damage and death can occur at 60%. Hemolysis is red blood cell death. The physiology of extravascular hemolysis. Hemolysis not in the vein. Spleen, liver, lymph nodes, and bone marrow. Hemoglobin is broken down by macrophages to heme and globin. AA of globin chains are recycled. Heme is broken down. Iron is transported by transferrin to the bone marrow or storage sites. Other parts are broken down into biliverdin, then reduced to bilirubin. Bilirubin is picked up by albumin and transported to the liver. Laboratory results in extravascular hemolysis. CBC shows decreased red blood cell count, hemoglobin, hematocrit, which is the ratio of cells to plasma, reticulocyte count increases, and polychromasia increases. Physiology of intravascular hemolysis. Hemolysis in the vein. Alpha and beta dimers are released into the plasma. Free hemoglobin binds to haptoglobin and is transported to the liver where it is broken down. Laboratory results in intravascular hemolysis. The CBC shows decreased red blood cells, hemoglobin, hematocrit, reticulocytes increase. Plasma results show decreased free haptoglobin, free hemoglobin, and lactate dehydrogenase increases. General laboratory evidence of hemolysis. The bone marrow shows erythroid hyperplasia or more red blood cell precursors in the bone marrow. The M to E ratio is shifted from 3:1 to 4:1 to 1:2. Peripheral smear shows increased polychromasia and possibly nucleated red blood cells and spherocytes. Pink to red shades in centrifuged plasma. Chemistry tests to determine intravascular versus extravascular. Classification of hemolytic anemias. It is important to classify the source of the hemolysis to detect the cause of the anemia. Anemia is a decreased hemoglobin which may be caused by decreased red blood cell count or decreased iron. Chapter 4 from the book, Hemoglobin Function and Principles of Hemolysis. Word key. Acidosis, the increase in the acidity of blood as in diabetic acidosis, caused by an excessive loss of bicarbonate as in renal disease. Alkalosis, increase in blood alkalinity caused by an accumulation of alkaline or reduction in acid. Allosteric, shape change. Amino acid, one of a large group of organic compounds marked by the presence of an amino group and a carboxyl group, the building blocks of protein and the end products of protein digestion. Complement, group of proteins in the blood that play a vital role in the body's immune defenses through a cascade of interaction. Cyanotic, having a blue tinge to the extremities. Hyperplasia, excessive proliferation of normal cells in the normal tissue of an organ. Polychromasia, blue tinge to the red blood cells indicating premature release. Hemoglobin structure and synthesis. Hemoglobin is the life-giving substance of every red blood cell, the oxygen-carrying component of the red blood cell. Each red blood cell is nothing more than a fluid-filled sac with the fluid containing hemoglobin. Every organ in the human body depends on oxygenation for growth and function, a process that requires hemoglobin. In 4 months, or 120 days, red blood cells with normal hemoglobin content submit to the rigors of circulation. Red blood cells are stretched, twisted, pummeled, and squeezed as they make their way through the circulatory watershed, all to deliver oxygen to the tissues of the body. The hemoglobin molecule, responsible for carrying oxygen, consists of 2 primary structures, heme and globin. The heme portion is made of 4 iron atoms in the ferrous state, as iron in the ferric state cannot bind oxygen. They are surrounded by protoporphyrin 9, or the porphyrin ring, a structure formed in the nucleated red blood cells. Protoporphyrin IX is the final product in the synthesis of the heme molecule. It results from the interaction of succinyl coenzyme A and delta aminolevulinic acid in the mitochondria of the nucleated red blood cells. Several intermediate byproducts are formed, including porphobilinogen, uroporphyrinogen, and coproporphyrin. When iron is incorporated, it combines with protoporphyrin to form the complete heme molecule. Defects in any of the intermediate products can impair hemoglobin function. The globin portion. This structure consists of amino acids linked together to form a polypeptide chain, a bracelet of amino acids. The most predominant chains for adult hemoglobins are the alpha and beta chains. Alpha chains have 141 amino acids in a unique arrangement, and beta chains have 146 amino acids in a unique arrangement. The heme and globin portions of the hemoglobin molecule are linked together by chemical bonds. 2,3-DPG is a substance produced via the Embden-Meyerhof pathway during anaerobic glycolysis. This structure is intimately related to oxygen affinity of hemoglobin and is explained in the hemoglobin function section. Each hemoglobin molecule consists of 4 heme molecules with iron at the center and 2 pairs of globin chains. The heme structure sits lodged in the pocket of the globin chains. Hemoglobin begins to be synthesized at the polychromatic normoblast stage of red blood cell development. This synthesis is visualized by the change in cytoplasmic color from a deep blue to a lavender-tinged cytoplasmic color. Of hemoglobin, 65% is synthesized before the red blood cell nucleus is extruded, with an additional 35% synthesized by the reticulocyte stage. Normal mature red blood cells have a full complement of hemoglobin, which occupies a little less than 1/2 of the surface area of the red blood cell. Genetics and chain formation of hemoglobin. 3 types of hemoglobin are synthesized in total red blood cell production: embryonic hemoglobins, fetal hemoglobin, and adult hemoglobin. Each of these types of hemoglobins has a specific arrangement of globin chains, a specific chromosome designation, and a specific number of genes relative to production. Chromosome 11 contains the genes for the production of epsilon, beta, and gamma, and delta chains. Each parent contributes 1 gene for the production of each of these chains. Therefore, each individual has 2 genes for the production of any of these chains. Chromosome 16 is responsible for the alpha and zeta genes. There are 2 genes on the chromosome for the production of alpha chains and 1 gene for the production of zeta chains. Each parent contributes 2 genes for the production of alpha chains and 1 gene for the production of zeta chains. Therefore, each individual has 4 genes for the production of alpha chains and 2 for zeta chains. Alpha chains are a constant component of adult hemoglobin. Each adult hemoglobin molecule has 2 obligatory alpha chains as part of its chemical configuration. Embryonic hemoglobin contains epsilon and zeta chains. Fetal hemoglobin, or hemoglobin F, contains alpha and gamma chains and begins to be synthesized at approximately 3 months of fetal development and remains at the majority of hemoglobin present at birth. At approximately 3 to 6 months after delivery, the amount of gamma chains declines and the amount of beta chains increases, making hemoglobin A the majority adult hemoglobin. Hemoglobin A2 and hemoglobin F are also part of the normal adult hemoglobin complement. The unique position of amino acids in each chain and the specificity of the amino acid itself are essential to the normal function of the hemoglobin molecule. Synthetic or structural abnormalities of the protein chains may lead to hemoglobin defects. Hemoglobin function. As previously mentioned, the principal purpose of hemoglobin is oxygen delivery. Additionally, the hemoglobin molecule is a structure capable of pulling CO2 away from the tissues and keeping a balanced pH within the blood. The hemoglobin molecule loads oxygen in a 1-to-1 ratio, 1 molecule of oxygen to 1 molecule of heme. Oxygen loading occurs in the oxygen-rich environment of the alveoli of the lungs. Hemoglobin has a high affinity for oxygen in this environment and becomes saturated with oxygen, forming oxyhemoglobin. The network of capillaries in the lungs makes the diffusion of oxygen a rapid process. As the oxyhemoglobin molecule transits through the circulation, it transports oxygen, unloading it to the tissues. In areas where there is a low concentration of oxygen, hemoglobin's affinity for oxygen decreases, allowing it to be released. As hemoglobin goes through the loading and unloading process, changes appear by the molecule. These changes are allosteric changes. The term allosteric relates to the way hemoglobin is able to rotate on its axis, determine the action of salt bridges between the globin structures, and dictate the movement of 2,3-DPG. The hemoglobin molecule can be either in a tense or in a relaxed form. When tense, hemoglobin is not oxygenated. 2,3-DPG is at the center of the molecule, and the salt bridges between the globin chains are in place. When oxygenated, the relaxed form is in place. 2,3-DPG is expelled, salt bridges are broken, and the molecule is capable of fully loading oxygen. Oxygen dissociation curve. The binding and release of oxygen from the hemoglobin molecule is demonstrated by the oxygen dissociation, or OD, curve. This curve is represented as a sigmoid shape. The distinct curve illustrates the unique qualities of oxygen dissociation and how the hemoglobin molecule and oxygen respond to normal and abnormal physiologies. When the hemoglobin molecule is fully saturated, it has all the oxygen it can hold and a high level of oxygen tension. As it travels from the pulmonary circulation to the venous circulation, it has more of an inclination to give up its oxygen in response to the oxygen needs of the tissue it is serving. In the normal curve, 75% of the hemoglobin molecule is saturated with oxygen, leaving 25% capable of being released to the tissue. There is a progressive increase in the percentage of hemoglobin that is bound with oxygen as the blood PO2 increases. In the lungs, in which the blood PO2 is 100 mmHg, hemoglobin is 97% saturated with oxygen. In venous circulation, in which the PO2 is 40 mmHg, 75% of the hemoglobin molecule is saturated with oxygen, and 25% of the oxygen is capable of being released when the hemoglobin level is normal. Two possibilities are demonstrated by this relationship. Depending on the need of tissues for oxygen, the hemoglobin molecule either holds onto oxygen affinity, or releases more oxygen as physiologic circumstances dictate. When referring to the OD curve, we speak about it in terms of having a shift to the right or a shift to the left. Shifting the OD curve means that physiologic conditions are present in the body that affect the relationship of oxygen to hemoglobin. In most cases, the hemoglobin molecule compensates by holding or delivering oxygen depending on tissue need. This compensatory mechanism can be demonstrated through the OD curve. When the OD curve is shifted to the right, hemoglobin has less attraction for oxygen and is more willing to release oxygen to the tissues. In a right-shifted OD curve at 40 mmHg, hemoglobin is 50% saturated but willing to give up 50% of its oxygen to the tissue because of need. When the OD curve is shifted to the left, hemoglobin has more of an attraction for oxygen and is less willing to release it to the tissues. In a left-shifted OD curve at 40 mmHg, hemoglobin is 75% saturated but willing to release only 12% to the tissues. Physiologic conditions that shift the OD curve to the right include anemia, decreased pH or acidosis, increased 2,3-DPG, and elevated body temperature. In the anemic state, the cells act more efficiently to deliver oxygen to the tissues, even though there are fewer red blood cells and less hemoglobin. The compensatory mechanism of the OD curve works adequately if the hemoglobin level is 8 to 12 grams per dl. For the most part, it is only when the hemoglobin level decreases to less than 8 grams per dl that symptoms start to develop. Conditions that shift the OD curve to the left are decreased 2,3-DPG, reduced body temperature, presence of abnormal hemoglobins or high oxygen affinity hemoglobins, multiple transfusions of stored blood, where 2,3-DPG is depleted by the storage process, and increased pH. Less oxygen is released to tissues under these conditions when 2,3-DPG is decreased. Consider this analogy for the OD curve. The OD curve is like a roller coaster. As you start to head up the incline, you hold on tight. As you roll down the hill, you are more willing to throw your arms up in the air and release or relax your grip. So it is with the right-shifted OD curve. Abnormal hemoglobins. Hemoglobins that are physiologically abnormal can produce conditions that are toxic to the human body. 3 abnormal hemoglobins are methemoglobin, sulfhemoglobin, and carboxyhemoglobin. Increased amounts of any of these abnormal hemoglobins in the bloodstream can be potentially fatal. Often, the production of abnormal hemoglobins results from accidental or purposeful ingestion or absorption of substances or drugs that are harmful. Abnormal hemoglobins also can be produced as a result of inherited defects. In the abnormal hemoglobin, methemoglobin, iron has been oxidized to the Fe3 state, which is no longer capable of binding oxygen. If methemoglobin levels are greater than 10%, individuals appear cyanotic or having a blue color, especially in the lips and fingers, and blood plasma has a chocolate color. Aniline drugs and some antimalarial treatments may induce a methemoglobinemia in individuals who are unable to reduce methemoglobin. Hemoglobin M, an inherited condition arising from an amino acid substitution, may also result in increased methemoglobin and cause cyanotic conditions. Carboxyhemoglobin, or hemoglobin bound to carbon monoxide, is often increased in smokers and certain industrial workers. This is because hemoglobin has an affinity for carbon monoxide that is 200 times greater than its affinity for oxygen. Thus, if carbon monoxide binds to hemoglobin, no oxygen can bind and no oxygen is delivered to the tissues. This can lead to carbon monoxide poisoning, whether deliberate or accidental. Although relatively painless, this process is reversible if caught in time. Sulfhemoglobin can be formed through exposure to agents such as sulfonamides or sulfa-containing drugs. The affinity of sulfhemoglobin for oxygen is 100 times lower than that of normal hemoglobin. It may be toxic at a very low level. Hemolysis. Red blood cell senescence, or death, is a natural process for red blood cells at the end of their 120-day lifespan. Red blood cell death occurs through rupture of the cell membrane, a process known as hemolysis. The contents of the red blood cell are released and returned to various parts of the circulation to be recycled in the process of forming new red blood cells. When red blood cell death occurs in an orderly fashion, the hematologic balance is maintained. Hemoglobin is kept at normal levels and the bone marrow maintains a steady production of red blood cells. If premature cell death or hemolysis occurs, a series of events begins to cascade, providing lab evidence that cells are dying faster than their normal 120-day life cycle. When cells start dying more quickly, the body's peripheral circulation begins an intervention process. Types and physiology of hemolysis. Hemolysis can be an extravascular or intravascular process. Of all hemolysis, 90% is extravascular and occurs in the spleen, liver, lymph nodes, and bone marrow, or the organs of the reticuloendothelial system. 10% of hemolysis is intravascular. Red blood cells are lysed directly in the blood vessel. It is imperative for a student to understand the process of extravascular and intravascular hemolysis. Physiology of extravascular hemolysis. Extravascular hemolysis is a well-established pathway in which red blood cells are destroyed and their contents are phagocytized. Hemoglobin is released into the macrophages where it is broken down to its heme and globin components. The amino acids of the globin chains are recycled into the amino acid pool, and the products of heme are taken through different pathways. Iron is transported by a transferrin, the transport protein, to the bone marrow or storage sites to be used in erythropoiesis. The rest of the hemoglobin molecule reacts with hemozygeinase, yielding a byproduct, biliverdin, which is reduced to unconjugated bilirubin. This bilirubin product attaches to albumin and is transported to the liver. In situations of increased extravascular hemolysis, the CBC will show reduced red blood cell count, hemoglobin, and hematocrit, and the reticulocyte count will be elevated as evidenced by polychromasia in the peripheral smear. Physiology of intravascular hemolysis. Intravascular hemolysis occurs as red blood cells are lysed directly in the blood vessel. The red blood cells burst, and the alpha and beta dimers of the hemoglobin protein are released immediately into the plasma. In the plasma, free hemoglobin binds to haptoglobin. The hemoglobin-haptoglobin complex is too large to be filtered by the kidneys, so it is not excreted, but rather transported to the liver, where it is destroyed and broken down. In situations of increased intravascular hemolysis, red blood cells, hemoglobin, and hematocrit are low. Also, free haptoglobin level decreases as it binds with free hemoglobin in the plasma. Reticulocytes are increased owing to the bone marrow trying to keep up with red blood cell production. This is known as erythroid hyperplasia. Additionally, lactate dehydrogenase, or LDH, a red blood cell enzyme, increases in the plasma as red blood cells are lysed prematurely. Conditions that may predispose to rapid and volatile lysis within vessels may activate complement, an efficient accessory to the hemolytic process. If complement is activated, as in the case of ABO transfusion reaction, proteins are introduced that cause additional damage to the red blood cell membrane. Laboratory evidence of hemolysis. Hemolysis can be determined in the lab by various methods. Most of these measurements are obtained in the hematology and chemistry laboratories by routine laboratory procedures. The evidence for hemolysis appears in the bone marrow, in the peripheral circulation, and in the blood plasma. The bone marrow shows erythroid hyperplasia, meaning an increase in red blood cell precursors and premature release of reticulocytes and young red blood cells. The normal myeloid erythroid ratio of 3:1 to 4:1 is shifted toward an increase in red blood cell precursors, giving a ratio of perhaps 1:2. The peripheral smear provides visual clues of hemolysis by showing an increase in polychromasia and the possible presence of nucleated red blood cells. For example, an elevated reticulocyte count from a hematology analyzer can be seen in the peripheral smear as polychromasia, or an increase in polychromatic cells. If antibody-coated red blood cells travel through the spleen is subsequently shears off the antibody, leaving the red blood cells in the spherocyte shape and diminishing the cell's lifespan. None of these visual indicators is likely to be observed in a normal peripheral smear. They are seen in a peripheral smear in response to a hemolytic event. In the plasma, the actual haptoglobin measurement is lower during a hemolytic event, owing to the excessive binding to free hemoglobin released from red blood cells during hemolysis. Once hemolysis has been determined, it is necessary to establish the type of hemolysis by analyzing the plasma changes. Many of the same laboratory findings present in intravascular events may also be present in extravascular hemolysis. However, intravascular and extravascular hemolysis can be distinguished by paying careful attention to laboratory data. Additionally, the spleen may become enlarged in extravascular hemolysis because damaged red blood cells may become sequestered in this organ. If the patient is experiencing intravascular hemolysis, hemoglobinemia, or free hemoglobinemia in the plasma, this may cause plasma to appear red or pink tinged. This color change resulting from hemoglobinemia can be observed after the blood sample is centrifuged. Hemoglobinuria, or hemoglobin in the urine, is the direct representation of free hemoglobin, or hemoglobin not bound to haptoglobin, being filtered by the kidney into the urine, causing urine samples to be blood tinged. This unusual finding is seen only in intravascular lysis. Hemoglobinuria may also be a byproduct of a direct intravascular lysis that may be complement mediated. Intravascular events may take 48 hours to manifest through laboratory results. Evidence of intravascular lysis indicates a serious condition that must be addressed promptly. Blood will be seen in the plasma and the urine, and the patient is at risk of a serious drop in hematocrit levels with 48 hours. Classifications relevant to the hemolytic anemias. The hemolytic anemias are classified according to many different schemes. In this chapter, we have classified the hemolytic anemias according to the site of hemolysis, either extravascular or intravascular. Many textbooks discuss the hemolytic anemias as caused by either intrinsic defects of the red blood cell or extrinsic defects. Intrinsic defects of the red blood cell relate to inherited deficiencies of the red blood cell membrane, hemoglobin structure or synthesis, or biochemical components. Extrinsic defects related to events that are secondary to red blood cell structure and function, but may still result in a hemolytic event. Regardless of the classification, it is paramount that the hemolytic anemias be recognized, evaluated, and managed so that life-threatening sequelae do not arise. Intrinsic red blood cell defects leading to hemolysis. Hemoglobinopathies, structural and synthetic. Red blood cell membrane defects. Red blood cell enzyme defects. And stem cell defects. Extrinsic defects leading to hemolysis. Autoimmune hemolytic anemia. Parasitic infection. Microangiopathic hemolytic anemia. Environmental agents, including venomous and chemical agents. Chapter 4 Summary Points. Hemoglobin has two main components, heme and globin. Heme consists of iron and the protoporphyrin ring. Globin consists of amino acid chains of specific lengths and specific amino acids. Alpha and beta chains are the two most significant amino acid chains. Alpha chains have 141 amino acids, and beta chains have 146 amino acids. Hemoglobin F is fetal hemoglobin. The adult hemoglobins are hemoglobin A, hemoglobin A2, and hemoglobin F. Oxygen delivery is the primary purpose of the hemoglobin molecule. 2,3-DPG is intimately related to the oxygen affinity of hemoglobin because it regulates the loading and unloading of oxygen. The OD curve schematically represents the saturation of hemoglobin with oxygen and the release of oxygen from the hemoglobin molecule under normal and abnormal physiologic conditions. Hemolysis is the destruction of the red blood cell before the end of its 120-day life cycle. Hemolysis may be classified as intravascular or extravascular, which relates to the site of hemolysis. Intravascular hemolysis occurs inside the blood vessels. Extravascular hemolysis occurs outside the blood vessels, primarily in the reticuloendothelial system. Hemolytic anemias may be classified by intrinsic defects of the red blood cell or intrinsic defects that affect the red blood cell. Review questions. The embryonic hemoglobins are Gower-1, Gower-2, and Portland. A person possesses 4 genes for the production of alpha chains. One condition that may shift the OD curve to the left is inheriting a high oxygen affinity hemoglobin. The basic components of the hemoglobin molecule include heme, globin chains, and 2,3-DPG. When a hemolytic event occurs, LDH is an enzyme that is increased. 2,3-DPG controls hemoglobin affinity for oxygen. When the iron in the hemoglobin molecule is in the ferric or Fe3 state, hemoglobin is termed methemoglobin. Hemoglobinuria can distinguish intravascular from extravascular lysis. Carbon monoxide poisoning results from carboxyhemoglobin intake. Fetal hemoglobin consists of alpha and gamma chains.